Hi everyone, thank you for coming to our presentation. My name is Shedivy and I'll be presenting on behalf of our groups from University of Wisconsin and Reed College. Today we'll be presenting our method for streamlining signaling pathway reconstruction. To start off, I just want to give a little bit of background on why pathway reconstructions are important for cell signaling. Most cellular processes are carried out by molecular interactions. And we can use omics data, such as genomic or proteomic data, to analyze these interactions by way of these signaling networks or pathways. This omics data is often most powerful when it's used in the context of a biological system, and this network-based pathway reconstruction method that we talk about is one way to do so. This technique of a pathway reconstruction has been used in many different areas, such as trying to identify mutated genes in cancer, or recovering differentially expressed subnetworks. We have a couple examples here that kind of highlight the success that pathway reconstructions have had. Here on the left, we have the HotNet2 method, and this method was developed to find mutated subnetworks in pan-cancer data. This method identifies subnetworks that consist of well-known cancer signaling pathways that contain multiple genes with rare mutations across multiple different cancers. By looking at this pan-cancer network analysis further, we can hopefully find new approaches for diagnostic and therapeutic opportunities. Here on the right, we have a graphical abstract of the TransposeNet method. And here, a pathway reconstruction algorithm was developed to map molecular pathways underlying the toxicity of alpha-synuclein, a protein central to Parkinson's disease. And here, the big idea is that they developed a method to humanize a molecular network of genes in yeast in order to prioritize human disease genes. We can kind of think of a general definition at a high level of a pathway reconstruction as just being organizing relevant genes and proteins in order to provide network context. They also offer a promising approach toward attaining a systems level understanding of cell signaling. Let's take a look at this pathway reconstruction task a little bit more in general. And if you recall from the last slide, we gave sort of a very high-level definition of what we mean by a pathway reconstruction, and that's just using some algorithmic approach to connect genes or proteins of interest within the context of some general PPI network in order to characterize some cellular response. Here we start with this background network or PPI of interest. We then want to select some subset of nodes, and those are highlighted here in yellow. And these can be either reception or transcription factors, differentially expressed genes. It really is going to depend on the context here. And this step takes domain knowledge to kind of understand, which proteins we want to study. We then want to perform the actual reconstruction in order to create this subnetwork. And this subnetwork here is actually running some reconstruction algorithm in order to create some kind of subnetwork, connecting these inputs that we've picked in step one. And I also want to mention here that we're kind of keeping things very high level right now. There's no notion of being connected or directionality or various node scores or edge scores, but we know that this task does involve reconstructing some kind of subnetwork given this bigger background PPI and some subset of nodes. That is the main general idea. Another thing I want to highlight here is that this task is often exploratory in nature. There's many different algorithms that exist, which we'll get into a few later, but they aren't widely adopted and they aren't widely known per se. And we want to kind of address this issue, and we'll get into this later in the presentation. Now that we've introduced pathway reconstructions at a high level and hopefully shown some of their promising applications, let's now dive into and take a look at what an actual pathway reconstruction analysis looks like on the ground. The first step will be to choose our inputs. This step will take domain knowledge in order to select some proteins or genes of interest in the context of a background network, and this is a similar step to the previous slide. We also want to make sure that we're taking care of the various attributes such as directionality, edge scores, or node scores. Once we have these inputs, we then need to actually choose the reconstruction algorithm to run. Here's just a list of some popular algorithms that are used. Right here at this step, we want to make sure that we're taking care of any algorithmic assumptions these algorithms will make in order to produce the subnetworks that we want. The next step here will be to actually format our data for the given method that we choose. And anyone with any data wrangling experience understands that this is a very time-consuming and tedious task. It can often be a challenge. This next step is also a practical challenge. Here, we actually just need to download the source code for the method that we choose. And here it's a challenge because we need to make sure that we have, the OS correct, along with any dependencies or runtime specifications correct in order to get the method up and running. This next step here is to select parameters to plug in with our inputs into the given method. This step will also take some degree of domain knowledge in order to choose a parameter for the given method to produce a subnetwork that's insightful for us. The next step will be to actually run the algorithm, given that everything else here is all in place. And then we can take the outputs that are produced in order to visualize them, like this example, or to run just various summary statistics on the given outputs. The big idea here is that at each point in this step-by-step work chart is that there are various challenges and things that can go wrong. And here there's various challenges that can kind of get in the way if we want to run multiple different algorithms on the same data. That is why we want to present a streamlined approach to this pathway reconstruction analysis. Here we take care of all of these practical challenges of formatting inputs, the OS and installing the method, and actually running the method and formatting the outputs. Here we just want a user to kind of look at what's important here, and that's choosing your inputs, choosing your method, and then choosing your parameters. And we'll get into this a little bit more on the next slide, but this is kind of our big picture approach here, is we want to streamline this technique in order for it to make it more scalable, more approachable, and more reproducible. Our prototyping framework is called the Signaling Pathway Reconstruction Analysis Streamliner, or SPRAS. Here we specify one type of input that's compatible with all supported methods, and this is kind of a universal file format. We can utilize the organization of a configuration file in order to specify all the different methods, parameters, and datasets that we want our workflow to run. The workflow itself is actually built on two major technologies, SnakeMake and Docker. Each reconstruction method is Dockerized in its own isolated container, which consists of a virtualized runtime environment, any of the method's dependencies, and the actual method's source code. These containers are compact and portable units which help standardize the way that methods are run within our workflow. They also provide very strong isolation from other containers and the underlying local system to help ensure that there's identical behavior across multiple platforms and multiple users. The workflow is actually managed by SnakeMake, and this is a formal workflow system that's built in Python. SnakeMake will handle all the input configurations that a user will specify and will handle all the different Docker interactions. After a given method is run, the SnakeMake will also handle any of the downstream analysis or output files. There's a couple different use cases that I want to describe real quick. And the first one is kind of from the perspective of a computational methods developer. And one of the great things about our workflow we're developing is that we can help support running multiple different actual pathway reconstruction methods on many different data sets. And this is kind of one of the goals of a computational methods developer. We can see that within that user specified config file, we can specify these various different inputs and the various different reconstruction algorithms. And SnakeMake will be able to handle all the other parameter combinations that we can run. We can then use the outputs that are generated to visualize these subnetworks or ensemble or evaluate them in any other specific way. I also want to mention here that SnakeMake offers a very good way to help kind of scale this workflow across various different clusters or cloud computing resources, which helps makes the transition a little bit easier if you're running, a very big analysis. Another use case comes from the perspective of a systems biologist. Say a biologist has proteins that are relevant to some kind of viral infection, and that can be identified with experiments in mass spectrometry, and that's just kind of depicted here in this Excel spreadsheet. They can take this data and they can run some analysis using a string protein-protein interaction network. And what they can do then is use our method to run Omics Integrator in order to compute and predict pathways that show these relationships within these proteins. In this example, we're finding peroxisomes that are important to certain infections, and using these subnetworks, we can design follow-up experiments that explore these relations further. For our ongoing work and upcoming features, we want to work on adding more reconstruction algorithms to our workflow, as well as address some back-end features such as ensembling and visualization techniques, as well as address this parameter selection issue, which I mentioned a couple slides back. As I mentioned before, we are in a prototyping stage, and so we really do appreciate any feedback that you may have in order to make this method broadly useful. We have our GitHub URL linked here at the bottom. Feel free to open any issues or pull requests with ideas that you may have. Thank you all for coming, and I'll answer any questions that you may have shortly.